Good morning, everybody, and thank you for having me here today. Wonderful, thank you. So as Paul mentioned, I'm going to be talking about cereal diseases and methodologies to control disease in the changing face of fungicide resistance. And we've had quite a bit of fungicide resistance develop within the cereal industry. So I'll be touching on what resistances we're facing, particularly here in South Australia, and then how we're going about presenting management strategies to industry. And we have a GRDC supported program that's run by the CCDM called AHRI, which is the Australian Fungicide Resistance and Extension Network. So as part of that program, we're developing strategies, methodologies, and resources for industry to help to manage fungicide resistance. So a bit of an overview as to what resistances we currently have in the grains industry. And you can see there's quite a long list there. The dots are then representing which state they are present in. They're not regionally specific within the state. And we'll be touching on today the barley and the wheat resistances and reduced sensitivities. You can see along the bottom here we've got 3 different categories, and this one here is a lab detection. We then have reduced sensitivity and full resistance, and these are indicated alongside the dots here. So this map's updated and available on the AHRI website, so check in and have a look to see if there's been any recent changes if you're interested. So I'm going to touch on, in the wheat, Septoria triticide blotch and powdery mildew, and in barley on our net blotches, net form and spot form of net blotch. All 4 of these diseases are quite prevalent throughout the state, and we get multiple reports every growing season from growers struggling to manage these diseases. And there's a range of factors that play into that. Fungicide resistance is becoming more and more of the questions that we're getting asked, but the disease life cycle and different management strategies that work for that specific disease are part of it too. So Septoria triticide blotch, we've got 2 different resistances and reduced sensitivities within that group. We've also got some field trials from a GRDC funded project looking at integrated disease management in the southern region. So I'll touch on some of the results from that and how we're using those to help inform growers of management strategies. So we have group 11 resistance in Septoria triticide blotch, which is the strobilurins. So in the cereals, we really only have 3 key fungicide groups available to us in terms of registrations. So group 11, this actually is at this stage lab detection only, so it's good news. It's originated from samples collected near a trial site in Millicent, which you can see in SA if you're not familiar with our state, is down here south of Adelaide, right down at the bottom. This is in our high rainfall zone, and we get heavy Septoria triticide blotch disease pressure there every year. So this mutation that was detected from a trial site has the G114.3A resistance gene, and mutation which we know confers resistance. Surveys that were then conducted in collaboration with the CCDM's fungicide resistance group through 2021, 2022 and 2023 showed that this was not widespread. We had the single case down in Millicent with a second case being found down there and 1 other sample in Tasmania. But outside of that, it wasn't detected. So this is really good news. We know it's there, but at this stage, really low levels. It's only been picked up in the lab, and we haven't had any evidence of that fungicide not working particularly well in the field at this stage. We also have the DMIs, or the group 3s, and we have reduced sensitivity to those. This is work that was done by New South Wales DPI prior to 2017 and concluding in 2017, looking at the G1 isoform of the CYP51 gene, and that affects some of our DMI chemistries, including tebuconazole, flutriafol, and propiconazole. And this is reduced sensitivity, so it's not full resistance. And what we see is that those different chemicals have different responses depending on location and which chemistry it is. The map that you can see here is the frequency when it was last surveyed by Andrew Milgate's group from 2017. And you can see that we do have this mutation throughout South Australia. We've got our yellow to red indicating that we've got prevalence of those mutations here. So we know we've got those reduced sensitivities in South Australia, and we're certainly looking at working around them, and local agronomists tend to have the best understanding of which particular actives within that group have the best efficacy. When we're talking about fungicide resistance management, we introduce this pyramid, and the idea of that is to talk about how fungicides are only one part of disease management. And that's really important with all of the diseases that we've got here. So at the bottom of our pyramid, and if you think about this like the old food pyramids that we used to have, that I think said we could eat a lot of carbs, which was great. I don't know where that's gone now. It seems to have changed. It's all keto and whatever else. But similar to that, variety selection is what our biggest choice should be, and one of our biggest drivers of managing diseases. So we've got variety selection at the bottom here. And so where possible, selecting varieties that are not susceptible to very susceptible to diseases that you have the highest pressure of in your region. We've then got our other disease management strategies, and so they play into the biology and the life cycle of the disease. So we're talking about things like our crop rotation, particularly if we've got a stubble-borne disease or a soil-borne disease. The stubble management is then obviously very important if we've got a stubble-borne disease. We have a lot of scenarios where there's very close rotations of crops, and we don't get good stubble breakdown in between seasons in our break year. And that can lead to disease epidemics in the following year. If we have the option to, we can look at sowing susceptible varieties later than our less susceptible varieties. So if you've got a longer season wheat and a shorter season wheat, and your shorter season wheat is the one that's more susceptible, can you push that back a little bit in your sowing program? That fits the maturity of the plant. But also for septoria, we know we get these ascospore showers that come in very early in the season. And then as we move past that, we're not getting so many spore showers. We're more reliant on the stubble in the paddock itself for that infection level. So once we've considered what other things we can do in terms of disease management, we're then considering our fungicide strategy and recognising that that plays one component of our management strategy overall. So using fungicides only when they're necessary and applying them strategically, rotating our modes of action, using mixtures, so not applying just one chemical on its own if that's available, and then staying within our label rates. So talking a little bit now about some of the trials we've been running in the last few years, and this is a project funded by GRDC, led by Agriculture Victoria, and in these trials we're collaborating with the Hartfield site group. And what we've been doing here is looking at the importance of variety selection on our disease development and yield loss of Septoria triticide blotch. And you can see across the bottom here we've got 6 different varieties ranging from Impala through to Lancer, and we've got an SVS resistance rating through to an MS, which was about as good as we could get in this particular growing region, and even still Lancer that was an MS was not particularly well adapted to this area. The blue here is the amount of disease The blue columns here were the amount of disease that developed during the growing season, and then our little dots are the amount of yield loss that each variety got. So we compared disease completely controlled versus allowing that disease to proliferate as much as it naturally would on that variety within the growing season. So we got up to about 30% disease severity, which is leaf area affected in Scepter, one of our S varieties, whereas you can see the disease levels in our MS and MSS varieties were sitting down under 10%, which is quite a bit lower. The yield losses that then were associated with that, we've got up to about 12% yield loss in Scepter, whereas we've only got about 4% yield loss in Lancer. So this is an example of how we can use our variety selection to then tailor the amount of disease that we're going to get during the growing season and subsequent yield loss. We also looked at the importance of timing of these fungicides, and we were targeting a seed treatment, a GS31 and a GS39 spray. And by doing this, we can see that an untreated control, we had no yield gain associated with that. Whereas when we move through to a 2-spray strategy in that season, we're getting up to about a 15% yield gain. Our single spray strategies at 31 and 39 is definitely still having an impact, and you can see that we've got between 8 and 10% yield gain there. We also know the importance of rainfall in a rain splash disease like Septoria triticide blotch, and we've been monitoring this by looking at having a modelling block each season where we're allowing the disease to proliferate naturally on a plot of an SVS variety, and then comparing that with the rainfall that we're getting during the growing season, along with taking disease ratings at every 2 weeks during the growing season. If you compare 2021, which was a drier season, with 2022, which we know was an exceptionally wet season at Hart in the mid-north of SA, you can see the amount of disease development over the course of the growing season doesn't really change or get to a significant level in 2021. And our variety trial that year had no yield losses recorded because the disease levels just did not get high enough. Whereas in a year where we've got that higher rainfall, in 2022, you can see that by the end of the growing season, we've got quite significant disease levels towards the end of the season. So having an idea of what kind of season we're expecting can also play into these decision-making processes with which fungicides we need to apply. So I'll now move on to powdery mildew. This is another big disease in South Australia and has certainly been growing in importance over the last few years. And unfortunately, we do have resistance and reduced sensitivity in South Australia, and we'll touch as well on some of the management strategies that we have available to us. This is one of the pathogens that we're starting to really struggle with control, and is highlighting how severe fungicide resistance can be on our farming system and what kind of impact it can have. So we have resistance to the group 11s, the strobulurins, and this is a data set produced by the GRDC and SAGIT projects that Trengove Consulting have been running in collaboration with CCDM and the Fungicide Resistance Group, who have been then running this screening. And you can see that in terms of across the state, the frequency of this particular mutation, we've got red, which means that it's at a very low frequency or not present, right up to 100%. And the importance of that frequency is we've got a diverse pathogen population. So you could potentially have 1% of the mutation present in your overall pathogen population and still have your fungicide appearing as sensitive and having good efficacy. And as that increases, you're going to get less and less sensitivity over time. So red is good, but you can see we've definitely got yellow, green and blue dots there, and they're dotted throughout our growing regions in South Australia. There is also reduced sensitivity in our DMIs, the group 3s. From the same project, there was a high prevalence of these mutations within South Australia. And we know that the level of efficacy then for the different chemical actives can vary quite a lot based on what pocket of the state you're in and what kind of strains you've got of the pathogen population there. And you can see this work here. These photos are from the CCDM produced by Fran Lopez, that when we've got the wild type compared with the resistant mutant, we've got a clean plant versus the powdery mildew still able to proliferate at that lower rate with propiconazole. So a really important one here in SA, and quite difficult for us to then work around, because that's 2 out of our 3 fungicide groups that we've got resistances and reduced sensitivities present in. I think another big factor with powdery mildew in South Australia is we talked about the importance of variety selection, and back to this pyramid that we work with. When we have a look at some of our commonly grown varieties in South Australia, we've got very limited options in terms of good resistance. A lot of our commonly grown varieties are VS, SVS, or an S rating. And that really is making it difficult for fungicides, even when they are sensitive and working well, to have an opportunity to work effectively. And this is because of the disease cycle and the ability of the pathogen to proliferate on an SVS variety with the right growing conditions. The breeders are working really hard on this now, which is positive, and we're hopefully getting more sources of resistance coming through. We've had Brumby, a variety from Intergrain, which you can see is green up there. When it first came out, it was resistant to powdery mildew. So that's really good news, but it is a little bit tricky at the moment. As a response to that, GPSA in South Australia worked with our local industry as well as GRDC and the APVMA to get some emergency permits through that allow some chemistries from other industries such as horticulture and viticulture for powdery mildew. So there's now 3 different chemistries that are available to growers. So that's really been helping them out in the previous season to get on top of powdery mildew issues. So how do we actually manage wheat powdery mildew? Variety selection, I think it's a matter of growers picking the best option that they've got, but also not expecting if they've got an S to a VS variety that they're going to get complete control. Paddock selection, I think we really need to be thinking more about our paddock selection, our rotations, our stubble retention systems, and what kind of disease risks they are creating. So we have a pocket where powdery mildew is quite bad on the northern York Peninsula, and in that area a wheat-lentil rotation is very common. The problem with that is that one lentil crop doesn't allow for a lot of breakdown of the pathogen on the stubble, and we're then going back into wheat stubble that still has active pathogen available on it in the following season. Preparing fungicide strategies, so rotating modes of action and combining our actives. We know from Sam Trengove's work that by combining actives, particularly combining multiple DMIs, we still can get some efficacy. Targeting our key timings, so canopy closure is a big one for wheat powdery mildew. It likes to sit on the base of the stem like you can see in the picture there, which enables it to then be a little bit protected from fungicides when they're applied. So the other thing for growers to consider is what kind of application type they're putting on. Anything with a lower water rate is going to have less opportunity to get down to that basal stem where it's really important to actually help control the disease. I'll touch now on the barleys, which is predominantly net blotches in South Australia. We actually have no spot form net blotch resistances currently in South Australia, so that's another good news story. But we do have several with net form net blotch, and that's our second one that we're really struggling to manage in South Australia. Both of the net blotches, we've got susceptible varieties with little diversity within the regions. Barley on barley, crop rotations are common. Early sowing is allowing more time for disease proliferation. High rainfall seasons are higher risk, and warm, humid conditions are worse for net form net blotch. The group 7 fluxapyroxide resistance, the SDHI, is prevalent quite a bit throughout the South Australian growing regions and a little bit into Victoria as well. And this is a map that I've produced from samples that we've received back from the CCDM from 2019 to 2021. We also have DMI resistance and reduced sensitivity, so the group 3s, and this is a snapshot of the work that from a field trip that Fran and his team and myself and my team did several years ago, which showed that the tebuconazole resistance was quite prevalent on the Yorke Peninsula. BASF have also reported this year that they have detected group 11 reduced sensitivity in the net form net blotches on the Yorke Peninsula as well. So we don't know a lot about this mutation at this stage. We know it doesn't confer full resistance, it's reduced sensitivity. It's at this stage picked up from their lab detection, so we don't know how that actually plays out in the field. So we need to monitor that over the next few years, because we're starting to really get into hot water here, because that will then be 3 of our fungicide modes of action that are then affected. Through our barley foliar pathogens project, which is a GRDC funded project, we collect samples right across Australia of net form, spot form and scald. And from that we conduct pathotyping, where we can see that we have quite a lot of diversity in the pathogen population in South Australia, as well as nationally for net form net blotch. So each isolate basically responds differently. And what this means is that this pathogen has a very fast ability to adapt to its environment and to the pressures that it's put under. So the resistance, if we have any in our host, so which genotypes we're growing, that becomes under pressure, as well as the fungicide applications. And that's why we've seen this move so quickly over the last 5 years, is because we have a sexually recombining population that already contains a lot of genetic diversity within it. And it's just important for us to remember that and try and protect varieties and fungicides as much as possible. So managing this, we're coming back to those same strategies where we're talking about the importance of varieties, stubble retention and paddock selection, and then considering our management strategies for fungicides, rotating modes of action, targeting key timings and monitoring paddocks. Spot form of net blotch, I mentioned that we don't currently have resistance or reduced sensitivity in South Australia. We do have resistance and reduced sensitivity to the group 3s next door in Victoria, so something we need to be aware of. And the group 7 SDHI resistance is prevalent in Western Australia as well. This pathogen has a lot less diversity in its genetics, and so we do have a pathogen population, but it's not quite as varied and as fast adapting and moving as the net form net blotch. So that's another thing that's good news. It doesn't mean that we should be abusing our varieties or our fungicides, but hopefully we've got a slightly lower risk of things moving as quickly as they have with the net form net blotch. So in summary, the fungicide resistance 5, and this comes back to our triangle. We've got variety selection being the most important, disease management, and then considering our fungicide options. And all of these resources are available on the AHRI website, and there's a fungicide resistance management guide that's available you can download with a lot more detailed information there too. So I'd like to say thank you. Obviously research doesn't happen without a huge group that are involved, and this certainly has had a huge group involved. We've worked quite closely with the CCDM over the past few years to understand what's been happening in South Australia. Thank you.
Thanks, Tara. We do have time for a couple of questions.
I have one question from the audience. Any update on cereal leaf rust resistance reported by Rob Park at the Wagga CPF event last year?
Yeah, I don't have any updated information on that at this stage, other than when they had a look back at that resistance and samples they've received over a long period, it looks like it has been there for quite a long time. So it's been prevalent in the leaf rust. In South Australia, we also have the alternate host, which allows for sexual recombination of leaf rust. So we do have a reasonable amount of diversity in the pathogen and the different strains that we've got here in South Australia. Other than we know that it's on the DMIs and that some of the chemistries are affected, I think we're seeing that in the field with the efficacy of some of our DMIs on barley leaf rust. I'm hoping that we get some more information at GRDC update time this year. Yep.
Tara, couple of comments regarding the 5 key parts for resistance management. Firstly, should we be trying to point out that people should be spraying preventatively or prior to a lot of disease development? Because I think often fungicides are timed a bit late in terms of getting on top of disease. And secondly, resistance testing, I think, is another important tool that we should be trying to incorporate as resistance builds. Yeah. We can't just assume certain products or modes of action are going to be effective. So I think we should be looking to add that to the list, or at least make people aware of that. We do it for weeds routinely, but insects and disease probably less so.
Yeah, I definitely agree with your second point there, and I'm going to take that as a statement rather than a question, and I think it's an important statement that really to continue to understand how to respond, we need the most up-to-date information, which means that we're getting consistent surveillance on what's happening within a region and where our fungicides are sitting for different pathogens. In terms of your first question, remind me what that was again. I can process one thing at a time, sorry. Spraying
early,
yes.
Before disease gets too established in the crop. I think some of these diseases are pretty hard to get on top of.
Definitely.
Even with a well-timed spray, but you can have the best product in the world if it's put on too late. That's not good.
Yeah, I think it's important to point out that a lot of our fungicides are much better in terms of their preventative effects rather than their curative effects. And so you're going to have a much better chance of controlling disease if you're ahead of the game. We saw an interesting situation on the lower York Peninsula from several growers and agronomists this year where they had applied Cestiva as a seed treatment for barley, for netform net blotch. The Cestiva had the resistance was present, and so the pathogen got started basically straight away rather than getting that period of protection from the chemical. The next application then went out at GS31, and in last year's growing season, that was too late. There was then follow-up sprays at GS39 and in some cases at about 50 to 60 as well. So what we saw there was in the SVS varieties particularly, that disease was then able to get away straight away. 31, we were relying then on the curative effect of the fungicides rather than the protective because the disease was already going, and that only then partially controlled it. And so because of the pressure of the season, with that good rainfall that we had over winter, it was allowed to then proliferate, and there were some pretty poor crops as a result of that. So I think, yeah, we do need to be prepared to go early. We also have to be careful that we're putting out fungicides when they're needed. So we're not putting out prophylactic sprays when we're not actually likely to have disease within our region. And I guess that's something we're looking at highlighting with the Septoria project, and in what seasons are we just seeing no disease development or disease development that doesn't cause yield loss. And we now have a Netform Net Blotch project, which we had a trial at Streaky Bay this year, and next year we'll have one at Hart and Streaky Bay, which hopefully will help to tell us some similar things for those regions. The high rainfall zones, obviously, you know you're going to get that rainfall every year and it's going to be consistent. But in our medium rainfall zones, it can be some years yes and some years no. And in our low rainfall zones, I think when growers are seeing disease there, they really have that question of what do we need to do with this? Do we need to spray? What's going to happen here?